I'd like to welcome Dr. Shikang or Scott Fan, Professor in Mechanical and Nuclear Engineering. He is a 2021 GFS Seed Grant recipient, and his project is titled Single-Cell Digital PCR Detection of Shiga-Toxin Producing E. coli with Point-of-Care Digital Microfluidics. Dr. Fan?
Yes, thank you, Jared. So, good afternoon. I'm very pleased and honored to present our work, which was supported by Global Food Systems Seed Grant. We are very grateful for this. I am Shikang Scott Fan from Mechanical and Nuclear Engineering. And today we will talk about this project titled STEC POCT, single cell digital PCR detection of shiga-toxin producing E. coli with point of care digital microfluidics. Although this title is quite long, but basically we are trying to implement a system called digital microfluidics and use that as a POCT platform and to implement a single cell digital PCR on this platform for the foodborne STEC diagnosis. And this project was carried by my group from College of Engineering and also the co-PI Jenfa Bai's group in the College of Veterinary Medicine. And today I will follow this outline. So I will give you some introduction and followed by some of our results and draw a conclusion and talk about some outcome and important impact. So first of all, STEC, shiga-toxin producing E. coli. So we know E. coli is a quite large and diverse group of bacteria, and most of them are harmless, but some of them can cause the severe illness, and some of them are transmitted by food. So for example, this foodborne pathogen, STEC, is the most notorious one. So when they are transmitted by food and taken by human beings, sometimes it will cause some very severe illness. So for example, like bloody diarrhea or the acute renal failure, like so-called HUS, which is life-threatening. So this kind of STEC will not only cause the public health issues like illness, hospitalization, and death, it also generate a lot of economic loss, which is a threat to the agricultural food. So for example, from the report from CDC and USDA, we know that in average recently, the United States, yearly we have about 265,000 infections per year, and that will cause 342 million economic loss. And within this, state of Kansas alone is suffer from 37 million economic loss because of this shiga-toxin producing E. coli. So apparently a rapid and novel STEC test is very essential, which is also our motivation in this project. Before talking about our solution, let's take a look at the current and conventional or the gold standard test of STEC. And based on the FSIS Microbiology Lab guidebook, to confirm the STEC contamination, there are many steps. So for example, we need to cultural enrichment. We do some PCR screening. We also do some immunomagnetic separation and followed by some cultural isolation after streaking. And then we will pick up the colony for the further agglutination study and streaking again, and then take the PCR and also some following steps before we can confirm the contamination, which can easily take more than 4 days to a week. So the reason behind this is because so this is one of the STEC I show here. This is an E. coli. So when we talk about STEC, it should carries the gene of certain serogroup like O157, 26, 45, 103, or 111, 121, or 145. So this is also so-called top 7 serogroups. And also at the same time, this bacteria should also carry the gene of shiga-toxin, like STX1 or STX2, and also intermin gene, which is EAE. Therefore, in fact, this repeating process we see, like repeating PCRs and this colony isolation culture, is because we want to make sure that the major virulence genes, which is this shiga-toxin and the intermin genes, are also carried by the adulterant E. coli serogroup, like this top 7. Therefore, so in the beginning, we do the bulk PCR. If we see both of the genes, we are not sure are they from the same identical bacteria cell or from multiple cells. So we cannot make sure that all the genes are coming from the same bacteria cell, which is STEC, or not. Therefore, the current gold standard is basically culture-based, which is labor-intensive, time-consuming, and definitely not ideal for trace the contamination, prevent outbreak, or even reduce the economic loss. So to overcome that, our co-PI, Dr. Bai, published their recent study. They use different ways. So they use a single cell digital PCR to do the STEC test. So this is cultural independent. So we don't need the culture to find the right colony and for the following analysis. We don't need culture, so that will save a lot of time. And this is single cell based, and we do the multiplex digital PCR. I want to use this figure to explain what happened here. So here, as you can see, we have several rectangles, and each rectangle represent one compartment or partition, or you can consider one well on a device, on a chip. So our goal, so in this single cell digital PCR, the process is that we try to make the cells they are separated in the individual wells, and each well will have only one or no more than one bacteria cells. So that when we do the PCR, so now after we partition in the single cells, when we do the PCR, now when we see the two genes, right? So one is the serogroup, the other is the virus genes. If they occur in the same well, then we know this must from the same bacterial cell, and we can confirm this is the STEC case. So in this case, the O157 and STX2 are on the single genome. However, on the right-hand side, although we also see the O157 and STX2, but most of them are from, and actually all of them are from from different cell, different bacterial cells. So we know this is a non-STEC situation. So meaning the bacteria will either carry STX2 or carry the O157, but none of them are STEC. So considering this, so if you imagine, if you do the conventional bulk PCR, you probably cannot distinguish the result between these two, because you will see essentially both of the signals in your PCR tube. So by using that, we can, Dr. Bai has reduced the diagnosis time within 1 day. So, but here we want to do even better, even faster. And this is labor saving because we have simplified the steps, and there is no like extraction, cultural isolation required. This is some of the current systems that can help us to do this digital single-cell digital PCR. So like here is a Fluidigm system, and on the right is a Thermo Fisher system. As you can see, they share something common, which is you probably need to start with a cartridge or a chip somewhere, and then you need to make the partitioning, make these small wells by their chip. You probably need a controller to do the partitioning, but you need another machine. So this machine is not small. This is like a refrigerator size. And you need another machine to do the PCR thermal cycle. And on the other hand, on the other side, you will see similar things. So you have multiple instruments. You need to have the skillful personnel to do the operation. So definitely multiple instrument and the still pretty large size. It is always usually this system is installed in a central lab, which limit the deployment for the on-site monitoring. So that is because there are still some bottleneck constraints of the microfluidic technologies that can use to do the liquid partitioning. So this is also one of the important thing we want to overcome in this project. So let me go it quickly for this part. So now we are going to the microfluidics or digital microfluidics part. After I explain the STEC, the gold standard, and the single cell digital PCR. So as you can see, most of the microfluidic has the channels, and many of the times you need to have some, like in this case, syringe pump to drive the liquid in the channels, which also you can see that in the previous case, in the previous slide, we can see those bulk instruments that are made to provide these purposes. However, in this case, in this project, we try to use the digital or the droplet-based microfluidics, or I call it DMF, which will drive the liquid by surface tension without using the channels or using the mechanical pumps. So that can reduce the system significantly. So show you a video and the picture to compare their differences. So apparently the conventional micro channel based, you can only flow the liquid continuously, but on the right hand side, we drive a 1 microliter water laterally without any channel walls confine the movement of the liquid. And also, one thing I want to point out is that on the left-hand side, we need a pressure. So that is why it is not easy to shrink the system to a small size. But on the right-hand side, we use surface tension, and we use the electric signal to control the surface tension so that it can be easily shrink the whole system size. Not to mention the programmability and the ease of fabrication. So a little bit about the theory, but here I just want to show you that by applying the voltage between the electrode, which is covered by a dielectric and hydrophobic coating, we are able to control the surface tension or control the contact angle. This is a simple equation, but I want you to focus on this gamma, which is the surface tension. So essentially, we can take advantage of surface tension. And as you know, surface tension is a super dominating force at the micro scale. So we want to use this efficient way to drive tiny droplets on the micro scale. So here is some of the videos shows you again, this is all like 1 microliter droplet moving around. And here is the real device looking like. So we have parallel plates. So again, I want to emphasize the structure is relatively simple. And we have some patterned electrodes on one side, and the liquid is sandwiched between them. So when we apply the voltage, we are able to drive the liquid to the more hydrophilic electrode. So later on, we will use the same device, but we will pattern the surface and make some hydrophilic holes that will be used for the liquid partitioning. So the first result shows you our implementation of the system. So this system contains a cartridge and also an instrument. So by using, because this is the controllability or programmability, we are able to do many different things. So for example, on the left-hand side, we can do the gene analysis. So in this case, we try to move a droplet, which is again like 1 microliter, 2 microliter, between 2 temperature zones. So now this instrument need to provide the functions of like thermal modules to control the temperature and electrical module to control the droplet motion. And also in some cases, in this case, if we want to do the PCR, we want to detect the optical signal. So we also provide the optical module to complete the job. So this is a way shows us the possibility to do the PCR or real-time PCR on this kind of cartridge. And this cartridge is also like on my hand. This is about 4 centimeter by 6 centimeter. By changing the electrode design, we can also do something differently. So this is basically an immunoassay. We try to capture our analyte on a magnetic bead, and before we add our substrate for chemiluminescence. So here, maybe I can show you a quick video like how to how to use this instrument. So this is our instrument. We put our sample, we put our cartridge in and inject some oil, which will be used for preventing contamination or evaporation. This is our magnetic bead. So the reagent will be programmed automatically and do the assay for us. We also have some magnets coming from the bottom to concentrate the magnetic beads for the extraction and washing. So after we do the multiple washing, the substrate will come from the other side, and now we are ready to do the final reaction. And the optical module on the top will record the chemiluminescence signals to complete the process. Okay, so this is the system development. Now the next question is, how do we do the digital PCR on this kind of system? So our scheme is to generate a micro well array. So think about that. If we can generate an array that composed of some hydrophilic well in the hydrophobic layer, so that when we pass the liquid through this microwell array, we will generate an array of a tiny droplet, which is used for the single cell partitioning. So think about that. This is a 2 microliter, and by patterning the hydrophobic and hydrophilic well, we can make the well generating this even smaller droplet, which is only 0.2 picoliter, so which has the 10 to the 7th differences. So that means if we use up all the liquid, we can generate 10 millions of the tiny droplets for the downstream digital PCR. So this is our goal. Again, this hydrophobic and hydrophilic pattern also take advantage of surface tension, which is very strong on the micro scale. So here shows you the result of patterning this well. So the well size is about 10 by 10 by 2 micrometer. So that gives us a 0.2 picoliter volume. So if you zoom in, you will see there are tons of this tiny wells. And we try to pass the droplet through the well array. So the droplet contains a rhodamine B, and also we put the E. coli, which is fluorescent labeled. So after we pass doing the liquid partitioning, we will see, yes, the liquid are partitioned in these wells, and also some of them captured with this fluorescence E. coli. So if you use so this is a fluorescence image. If you use a bright field, you can also see some of the wells have only liquid. Some of the wells contains also the single cells. And the median is an oil to avoid any evaporation, which will be ready for the PCR. Okay, so however, before do the PCR, we also try to do the on-chip PCR testing, which we use a bigger droplet. Instead of using that 0.1 picoliter, here we still use the 2 microliter. We just want to make sure that the PCR is performed correctly. So this is our thermal cycle, and we use the conventional kit, which and the probe, which will show us the 0157 using the FEM probe, and STX2 use the VIC probe. So after the 40 cycles, we can see in the STEC case, we will see both of the genes amplified. And in the no template control, we don't see any fluorescence dye. So meaning that the thermal module is also functioning well. So now we are ready to test this digital PCR. So this is the result of testing the digital PCR. So in this case, we first try to do the quantification of E. coli. So we put the E. coli, which has the 0157 positive, and use the same FEM probe as I showed before to see the result. So here is like no template with 0 concentration. This is 20 cells per microliter. You start to see some of the fluorescence dot. And if we increasing the concentration, you will see more dot. And by calculating the dot numbers, by using this equation, so this equation will show us the K is the positive number of partition, N is the total number of the partition, and the lambda will be the number of the template per partition. So that will give us the concentration. So digital PCR is the absolute quantification technique. So we don't need the standard curve. So this is what we get by analyzing this K over N. We can find this concentration, and which is a chord which agrees with the concentration we put in. So this is the first demonstration of the quantification, and then excuse me, we do the STEC and non-STEC detection. So here we have 3 groups. One is the STEC, the second is non-STEC, the third is no template control. So here the concentration is this, and as you can see, after we do the PCR for the STEC case, you will see the same point. You will see the 0157 and EAE signals, but for the non-STEC, we only see the 0157, and no template, we don't see any signals. So in the future, we will keep working on that and do the quantification and even testing the food samples. So the conclusion is that we developed a DNF system for STEC POCT. We have the instrument, we have the cartridge that contains a microwell array for single cell digital PCR. Including the enrichment time, we can finish the assay within 10 hours. and the sensitivity shown before is 20 cells per microliter. And this is one stop. We don't need to transfer any samples, so this instrument will provide the thermal, optical, and electrical signals for us. And the size is compact that it will be suitable for on-site monitoring. So in the next step, we'll be doing the more systematic study and for the beef samples. So talking about the outcomes in terms of students, we have postdoc, graduate student, undergraduate student involved and educated, and also the student also received several awards during this time. And we also did the grant application. We submitted two. So both of them are submitted to USDA NIFA program. So the first one was declined. The second one is under review. And during the period of time, we also established some collaborations, including some local company and also more group members that in this case, we try to do the similar digital PCR by using another material, which is hydrogel provided by Dr. Sung from grain science department. And in the future, we believe this kind of technology will be significantly helping to reduce the economic loss, especially for the beef industry in Kansas. And also that will enrich human health by the timing diagnosis to reduce the medical treatment. And finally, we hope this kind of POCT will be reliable, cheap, and easy to use. That can be used for the entire beef supply chain. Will be facilitating the early detection to stop the contamination and the outbreak. Job creation, of course, we will facilitate the industry of diagnosis, but at the same time, we also anticipate to foster the microfluidic diagnosis industry. And 5 students are trained, which is also the potential workforce. And we will also, we are working on the journal articles, which will be published soon. Okay, so with that, again, I want to acknowledge the financial support from Global Food Systems seed grant, and thank you for your attention.
Thank you, Dr. Fain. And if anyone that is listening to or watching this has any questions or wants to know more about this project, you can reach out to Dr. Fain.